It's my pleasure to introduce, and Pedro, I promise you will give you your full half hour to educate us this morning. Pedro Rioseco comes to us from the Advancing Eco-Agriculture, known from John Kemp, who's well known with respect to a really holistic approach, a very regenerative approach to soils and to health and agricultural production. Pedro was trained in genetics. I didn't tell you I was too. I'm a geneticist as well. And it's funny as how we move to where the kind of the need is in the world at the moment. And Pedro has had a lot of experience then in cropping, in looking from an agronomic sense at changes and shifts, and he's had different professional experiences, but has moved into this whole arena, which I think is not emerging, but as we're learning about and should emerge, as far as immunity that plants can get through better nutrition to the kinds of diseases and insects that want to attack them. This is critical, I think, as we look for, it's not just getting biodiversity in our cropping system, it's getting plant health. And with that, Pedro, we're very anxious to hear you present to us and educate us on this very important topic. Pedro Rioseco.
Thank you, Tim. It's a real pleasure to be here with all of you. And I hope you're just as inspired as I am hearing Scott, Kurt, and Dale. I think that was a wonderful set of presentations in seeing what Regen Ag can do for you. But also there's another conversation to have about the impacts on health, on human health. There's a conversation to have about growing crops and using food again as medicine. And it all starts with soil health and plant health. I've been invited to tell you a little bit about the connection between plant health and plant immunity. I'm going to begin by saying or repeating what the panel said about conventional agriculture and where have we gone there? You know, for the past 170 years, roughly, we've been instructed to think about nutrition in terms of ions, and whether that is partially true, that's not the way nature works. That's not the way plants function, or the sole way of that plants function. We all know intuitively that nature works in a different way. It's a system, and it's a very complex interconnection that makes this ecosystem work. Nobody is fertilizing a forest. Nobody's fertilizing the Amazon, yet plants thrive. Why? Well, there's something to do with microbiology. Dr. James White's work in rhizophagy, rhizo from root, sphagy from eating, he has demonstrated that bacteria and other organisms live inside plants and help them get the nutrition and defend against other pathogenic organisms. I'm not going to go into a lot of in depth into this topic, but I wanted to show you that he was able to demonstrate that down there in the left corner, two visuals of root systems that without the presence of biology, this is really critical. I want you to understand that without biology, without bacteria, root hairs cannot grow. They just simply can't grow without bacteria. And we all know that root hairs are in charge of providing some of that moisture and nutrition to plants. And not only that, they provide secondary metabolites and regulate hormones. So why is this relevant? Because he was able to demonstrate that plants can take in full bacteria, stripe them from their cell wall, and obtain more complex molecules as nutrition. That makes the system way more effective, more efficient, and also provides the raw materials for secondary metabolites and hormones. Now we take these step further into these endophytes that we know now they live and communicate with plants, and plants are able to take them in, feed them, spit them out into the soil again, and continue this cycle where they will help mineralize those nutrients for the plant, and it's a perpetual system. But plants are intelligent enough to select for those populations that will work with them and take them into the next generation. For some of you, probably the term epigenetics is not new. But for those who have never heard it, it's the expression of the genes. So you may have identical genes, but organisms express differently depending on the environment. So it's the expression of those genes by the environment. And when you have these endophytes, you have a more robust and resilient plant compared to plants that have been completely stripped from those associations, and that happens with a lot of breeding. So I brought this topic because we have to consider when we are selecting varietals or rootstock, if they have been bred in a complete sterile environment, you may have lost some of those connections. And I'm not gonna go into GMOs, but there's also a negative impact in the association with these microbes. Practices that would enhance microbiology. In the previous presentations, they spoke about a lot of how to protect those microbes or to protect the soil and build soil, right? There's a lot of practices in regenerative agriculture, including keeping root systems alive for the most part of the year, as long as you can, integrating livestock when applicable, and so forth. So in summer here, you may have temperatures, soil temperatures reaching up to 140 degrees or more if you have bare soils, compared to potentially during that same day, if you have a light cover crop or a light mulch, you may have temperatures of 90. There's a big difference, and microbes are known to die or stop when they reach 110, 120 degrees. So basically, when we use these conventional practices and keep the soil bare, you're pretty much presenting a situation where biology may just die completely in the first 2 to 3 inches. And if on top of that you add compaction, where you may have compaction at, let's say, 5 inches, then all your remaining environment for that biology to thrive may be in just 1 inch. That's how fragile we can make our own systems, versus protecting that biology, helping the soil have the aggregation, that gas exchange, which can make our systems more resilient. Scott spoke about the intelligent use of tillage. Tillage is a tool that can be used to help grow, and it has been demonstrated that intelligent use and with knowledge and intention, you can still till in maybe 2 to 3 inches, and as long as you don't destroy all the biology down to 6 to 8 inches, that biology will recolonize. So not all the practices, we have to think about this as not necessarily dogmatic. There are tools that can help in our systems to continue regenerating. There's also a conversation to have about resilience and climate resilience. There's both on the water conservation and water efficiency, just like Scott very well presented, and the nutrition associated with it. There's all these conversation about microbes being able to mineralize all those nutrients. He was talking about reduction of inputs because he was using what he called the compost, or reintegrating those crops into the soil, where the microbiology works for you, fixing nitrogen. There's an abundance of nitrogen above our head, and the microbes are able to bring that back into the soil, alongside carbon, alongside many other things that the system, complete system may work. And microbes can provide, so let me say this other way. Plants with association of microbes and biology, particularly mycorrhizae, can explore way more surface than roots can, and access some of these smaller pores where they can get both moisture and nutrition during drought. There's evidence that when you see soil, that in our touch and our if you just grab it, it looks like dust, but yet has up to 60 or 70% moisture in that biofilm close in proximity to the colloid. And microbiology is able to access that and provide it to the plant. Control deficit irrigation or stressing plants. There's a conversation to have about concentrating these secondary metabolites through stressing the plants, and it may work. There's some studies shown that if you have a plant growing, right, and then you stress it, you will dehydrate it and concentrate those nutrients or those secondary metabolites or the essential oils, in other words. But the story is incomplete, because it has been proven if you continue providing adequate nutrition and the optimal conditions to the plant, that curve will reinvert, and now you have more biomass and more concentration of secondary metabolites. It's just a conversation we have not had because of the reductionist approach of analyzing this in a lab. Now we're going to get into the fun part when it comes to nutrition, and I want to talk about nutrient integrity. USDA has a study of apples and concentration of nutrition on an apple today and 70 years ago. And it turns out that now we need to eat 3 apples in order to get the same nutrition. I'm pretty sure that most of us have had that nostalgia of remembering how amazing sometimes some fruit tastes. Nowadays, it's hard to get a tomato that tastes like a tomato. And so forth. Pears, my favorite pears, and the taste and the aroma that you can have when they're grown in combination with microbes, it's outstanding. Why? Because these microbes, as we saw before in the rhizophagy, they are responsible for providing the building blocks for all these secondary metabolites, all these flavonoids, carotenoids, sesquiterpenes, etc. That it's just, we have the most sophisticated measuring tools in our tongue and our nose to detect nutrition. And all we need to do, when we taste something that's wonderful, because our body recognizes that has that nutrition, we just automatically like it. And that has to do with nutrient integrity. We're not the only ones. Insects, there are studies that both bumblebees and bees are very selective of which flowers they wanna get pollen from and stop on, and they can detect through electromagnetic waves which flowers have a higher nutrient density, and they will never stop in certain flowers that have a very low Brix level or sugar content. They know. And they recognize it through electromagnetics. They can see it like we see neon lights in the night. And this has to do with the vibration of those molecules. And not all nutrition is the same. And we don't need the same every single time. Just like mothers may have cravings when they are pregnant, same thing, plants will have different requirements at different stages of development. At AEA, we call them critical points of influence. There are these stages of development where there's a switch between the vegetative growth and the reproductive growth. and has to do with hormonal balance, but also nutritional needs. And we can influence these with nutrition and also support it to have a better quality product and avoid certain deficiencies during the growing season. An example is you need, during cell division phase, you need a lot of calcium. All the cells that are gonna be divided are during this period. And then you get into the fruit field, and that's when you will no longer be able to influence the number of cells. All you can do is fill it with nutrition and sugars, and for that, potassium could be key. But if you don't have enough calcium, imagine this as a bucket full of balloons. So you can have a couple of buckets. In one, you may have 3 or 4 balloons filled with water, and in the other one, you may have 100 balloons. You have way more capacity of filling that and sizing fruit than you would in these other ones that may burst at some point. And that's when we have issues like with tomatoes, you want the end rot and so forth. That's because of that calcium deficiency, but it's very specific during cell division. You may add calcium later on and it will do nothing. So this is what we mean with critical points of influence. And then we have to also talk about the nutrient interactions. So there is a relationship between potassium and manganese. There's also a relationship between phosphorus and zinc, antagonism between cations and as well ions among themselves, et cetera, et cetera. So when we design a nutrition program, we need to consider this, or when we're analyzing a soil test, a SAP test, or whatever tools we're using to measure our approach, we need to consider the interaction between these nutrients. It happens both in the soil and as well inside the plant. So if we are upfronting a lot of potassium, because that's the only time that we will have an opportunity to put it out there, we may be antagonizing all that calcium when the plant needs it the most. And then we're wondering, why are we having all these quality issues? Nutrition can also influence the hormonal balance of plants. There is a very tight relationship, a tight connection between some nutrients and hormones. The nutrients associated with auxin and vegetative growth are nitrate, potassium, and chloride. And the nutrients that will promote cytokine dominance, or the reproductive hormone, are manganese, phosphorus, ammonium, and calcium. So by managing nutrition differently, you can either promote more vegetative growth, or you can promote more reproductive growth. That will depend also on your crop. If you're growing alfalfa or pasture, you may not want to focus too heavily on those that will promote the reproductive energy of the plant, and so forth. And now, I want to connect these dots with plant immunity. Just like us humans, that if we know that if we are well nourished, and we have heard that we are well nourished, our immune systems would be stronger. And it's the same for plants. Plants are capable of completely resisting diseases and insects when they have the adequate nutrition and the right microbiome, right abundance and right populations, like I spoke before. So it's not only a matter of chemicals, it's a matter of the whole system, and it works in conjunction with biology. Nevertheless, we can influence through our management practices how the plant will respond, and we can activate that SARS and ISR pathways that can help plants resist disease and insect attack. At AEA, we developed what is called the plant health pyramid. You may have seen this. John has spoken about the 4 levels of plant health, where the first 2 are passive, and the second 2 are active immune system. What I mean by that is the first 2, you remove the source of food for pathogens and insects. I'll give you an example shortly. The second two are active immunity. They can kill on contact, and this is when the plants, with the association with biology, can produce these secondary metabolites that we all know as essential oils, and we know how powerful they are and the properties they have, antiviral, antifungal, antibacterial, etc. We're going to dig a little bit deeper into these. So first, complete photosynthesis. If you have a plant that has a higher degree of efficiency, of photosynthetic efficiency, you may start resisting some of the soil-borne pathogens. So the moment that you remove partial carbohydrates, simple sugars, those pathogens no longer find a food source. And in order to achieve these, you need adequate levels of magnesium, manganese, nitrogen, iron, and phosphorus. This doesn't mean that you need to add more. You just need to make sure that the plant has sufficient levels. And SAP analysis is a really good tool to understand that, because just like you would go to the doctor and he doesn't ask, what did you eat? What was your plate looking like? He goes and have some blood tests drawn, and he wants to see exactly what's going on with you and not so much of what you're eating. The same principle works. It's good to know what the soil looks like, but that doesn't always tell exactly what happened and what the plant was able to absorb. I'm not going to go into the detail of why are those elements important for the sake of time, but those are critical for photosynthetic efficiency and capacity. Now we come to the second level, where plants are able to, those sugars, now they add a nitrogen element, and they convert this into amino acids, peptides, complete proteins. Once the plant has those elements, those enzyme and enzyme cofactors, to be able to convert these simple sugar forms into complex proteins, now you're starting to resist simple digestive insects, such as aphids, whiteflies, and so forth. Why? Because just like you and I cannot go out there and eat pasture because we don't have the enzymes to break down cellulose, the same way, the same principle applies for insects that they don't have the enzymes to digest complex proteins. Converting that nitrogen, when we use nitrates or urea, we need different elements to convert that into a more complex form of nitrogen. And as you go on in the chain, more enzyme and enzyme cofactors and nutrients are needed. So when we talk about that simplistic approach and reduction approach of NPK only, you're missing a lot of these, and that's why plants have become very susceptible to disease and insect. And now you rely on pesticide, on other means of control, because your plants are pretty much starving. This is from Tom Dijkstra. I found this fascinating. Most likely you have heard that Brix levels are associated with insect resistance. And the higher the Brix level of your plant, the more the level of resistance that you achieve in different insect categories. Even though it's true in a way, it's a broader conversation. It's not only about sugars, but being able to convert all those proteins, have complete proteins, have the enzymes, et cetera, but it's a good measure if that's all you have available at the moment. But what I'm just gonna say is that as you increase the level of health in your plant, you will start resisting these different insects. So if you're going through your orchard or your field and you find sucking insects, you most likely have a Brix level of 7 to 9. And once you increase the level of health of that plant, they will no longer be interested in those plants because they simply cannot feed on them. Then you reach to the fourth level where you increase the lipid content. Once plants have abundance of energy, they can exudate up to 25 or 70%, 25 to 70% of those photosynthates they can exudate through the roots to feed biology. That's how That's how nature designed it. That's why it's so important to look at soil health, because if plants are willing, and they're very intelligent organisms, if they are willing to invest up to 70% of their photosynthase to feed that biology, that means it's important. Why they do this? Because as they have abundance of nutrients, minerals, and so forth, they now can start converting those proteins or those simple molecules into lipids, store it, but most importantly, they have this waxy sheen, wax layer in the leaf surface that can help regulate temperature. So you may have air temperatures of, I don't know, I'm gonna say hypothetically 85, where the leaf temperature may be 65. Because when you get to 78 degrees Fahrenheit on the leaf temperature, not ambient temperature, but leaf temperature, you shut down photosynthesis. And when the plant shuts down photosynthesis, now it starts to consume itself in order to cool off and to have energy. And that's a situation you don't want to be. You raised ammonium levels, et cetera, attracting insects. Another thing is Callahan was able to show that plants, their neurological system works through the wax layer. That's how they communicate. That's how the electricity travels. So if you have a plant that doesn't have enough lipids, doesn't have that waxy sheen, then simply it doesn't function properly. And then finally, you get to that fourth level where, with an abundance and the right population of biology in the soil, you can start generating those secondary metabolites, essential oils, where the plant gets its full protection. And lastly, I'm going to talk about SAP analysis, the tool that up to this day has proven to be the most accurate in understanding plant needs, requirements, and how to manage better nutrition. This is an example where SAP analysis, how does it work? Well, you have old leaf, new leaf. It's a differential test. The reason you do this is because of mobility of nutrients. So it's very different when you are trying to analyze, say, potassium that can be mobilized from old leaf to new leaf versus, say, iron or manganese that are a little bit more less mobile. The reason to do this as well is because, different from, let's say, tissue test, you're seeing what happened already in the past, what's accumulated, and you don't know the form of that nutrient. In order for these metals to be available and bioavailable and the plant being able to utilize them, they need to be in the right form, in the right reduced form, and also the right chelation. That's a reason sometimes you may have experience when you have tissue tests showing adequate levels of iron, adequate levels of manganese, for instance, and yet you see it visually that the plant is still showing symptoms of deficiency. Plant SAP analysis would show you the element in the right form that's running through the veins of the plant and what the plant is needing in that moment in time. Yeah, I think that's it. Thank you so much for your attention.
We have 1 minute for questions, and I know we have 400 questions. I have them myself. Anybody? We'll take one. There's one down here, Zion, if you would, please.
Is there any particular time during the growing season when you do plant SAP analysis, and how many labs are available in the United States?
Great question. We recommend to do it, especially you're starting, depending on your crop. You can do up to every 2 weeks. And that will give you a trend that will help you understand the movements and the requirements of nutrition and what is the system providing for your crop. Later on, you may focus, once you have a robust database, you may focus on those critical points of influence to try to manage better your nutrition. You can start as early as bud break in case of some crops, all the way to harvest. That's what will give you the best information. Labs in the States, the best one is in the Netherlands that we know of. It's called Nova Crop. There are a couple in the U.S., New Age and Apical, that you may want to also check out. Those three would we have most experience with those three. Still, Nova has shown least inconsistencies.
Pedro, we very, very much appreciate this. We get to be with them this afternoon for sap analysis out in demonstration, and this is an opportunity to continue this conversation and questions. Thank you very, very much.